Imagine your DNA is basically an encyclopedia of 3 billion letters.
Right, a massive stack of books.
Exactly. And we know that just one single typo in that massive stack can cause, a devastating genetic disease.
Yeah, just one altered letter.
But here is the problem we're facing today. Thanks to a decade and a half of really advanced genome sequencing, we haven't just found one typo.
No, we found millions.
Millions of them, just scattered all across the human population. And up until right now, not even the world's most advanced artificial intelligence could look at most of those typos and tell us which ones are deadly and which ones are, totally harmless.
Yeah, and in the medical field, we actually call these VUS, or variants of unknown significance.
Right.
It's incredibly frustrating for a patient, right? You get a genetic screen only for the doctor to point to a mutation and essentially say, well, we see a change here, but we have absolutely no idea what it does.
Which is terrifying for the patient.
Yeah.
And that brings us to the mission of today's deep dive. We are looking at a really groundbreaking solution to this exact problem. We're looking at a new paper detailing an AI model called eSIG-Net. The goal here is to understand how this specific model can zero in on a single microscopic genetic mutation and accurately predict if it's going to destroy a critical biological relationship.
And spoiler alert for anyone listening, this is a task that has historically just humiliated some of the biggest names in AI.
Oh, absolutely.
Yeah.
But to understand why this is such a notoriously difficult problem, we have to look at the biology first, right? Like, what is a missense variant?
Right. So a missense variant is when a single DNA mutation causes just one amino acid to change in an entire protein sequence.
Just one.
Just one. And proteins, as we know, they don't just float around aimlessly. They're like the molecular machines of your body.
Okay.
And they do their jobs by physically interacting with other proteins. We call these protein-protein interactions, or PPIs.
So it's a relationship. If a normal protein interaction is, say, like a highly specific key fitting perfectly into a lock, a missense mutation is like altering just one microscopic ridge on that key.
That's a perfect analogy because sometimes, you put the altered key in the lock and it turns just fine. The change didn't actually matter.
Right.
But other times, that one tiny altered ridge completely jams the entire mechanism.
And that jamming effect creates what you guys in computational biology call an interaction cliff, right?
Exactly, and an interaction cliff. It's very similar to an activity cliff in chemistry. You don't see a gradual, say, 10 or 20% decline in how well the protein works.
It's not a slow fade.
No, it's completely binary. One single amino acid changes, and the entire signaling pathway just falls off a cliff. The relationship is utterly severed.
Okay, but if you're listening to this and you follow tech at all, you're probably, like, screaming at your device right now.
Yeah.
Wait, didn't AlphaFold essentially solve biology?
Right. Everyone asks that.
Why can't we just use existing famous AI tools to look at the 3D shape of the mutated key and the lock and just, visually check if they still fit together?
I mean, it is the most logical question to ask, but the data shows that structure-based models completely faceplant on this specific task.
Really faceplant?
Oh, totally. The researchers behind eSIG-Net actually tested this using AlphaFold-derived tools, like FoldDoc.
Okay.
And when FoldDoc predicts the 3D structure of the normal protein, the wild type, and then predicts the structure of the mutated protein, the AI sees them as virtually identical.
Wait, how identical are we talking? Give me a sense of the blind spot here.
Well, in instructional biology, we use metrics like TM score and RMSD to measure how much two 3D shapes overlap.
Right.
And the AI scored the mutant and the normal protein with an average TM score of 0.84 and an RMSD of 0.92 angstroms.
Which means what in plain English?
Without getting bogged down in the math, those numbers mean that to the AI's eye, these two proteins are identical twins.
Wow.
Because the overall microscopic 3D structure hasn't collapsed, the AI gives them almost identical interaction probability scores. It is completely blind to the interaction cliff.
So AlphaFold is looking at the macro shape of the key and saying, yeah, looks good to me, completely missing that the microscopic ridge is going to jam the lock.
Exactly. But it's not just structure models that fail. Standard protein language models, like ESM1B or Descript-T, they also struggle heavily with this.
Oh, interesting. Why do the language models fail?
Because standard language models are trained to look at the entire sequence of a protein to derive, like, a general context.
Right, they read the whole book.
Yeah. So when you feed them a sequence of 1,000 amino acids and only one has changed, that single mutation just gets drowned out in the noise of the overall data.
Okay, so let's do a quick recap for you listening. The problem we're facing is that a single typo causes a massive downstream disaster. But our current tools either zoom out too far, see the 3D shape, missing the tiny detail,
Right.
or they read the whole text, and the typo just gets lost in the noise. So my question to you, the listener, is this. If you were designing an AI, how would you force it to care about one single changed letter in a book of thousands?
That is the exact hurdle, which, forces us to rethink the architecture entirely.
You have to build something new.
Right. If you want an AI to catch the anomaly, you can't just teach it to read. You have to teach it to play a hyper-focused game of spot the difference.
And that takes us under the hood of eSIG-Net. This AI doesn't just look at the mutant and guess if it works.
No, not at all.
It operates by hyperfixating on the discrepancy between the normal version and the mutated version. But before it can look for differences, it has to translate the biology into something a computer can actually understand, right?
Exactly. AI doesn't read biological letters, it reads math. So the first step eSIG-Net takes is converting the protein sequences into this massive 573-dimensional mathematical fingerprint.
Wait, hold on. 573 dimensions. That sounds like absolute overkill.
It sounds like a lot, yeah.
What physical reality are we actually measuring with that many numbers?
So it's doing 3 distinct things to build a comprehensive profile of the protein. The first part is pretty simple. It's amino acid composition. There are 20 standard amino acids, so the model just counts the frequency of each one. That's your first 20 dimensions, just the raw ingredients.
Okay, an ingredient list makes perfect sense.
But ingredients don't tell you how a protein folds, So the second method is called the conjoint triad.
Conjoint triad.
Right. Proteins fold based heavily on 2 things. How bulky their molecules are, and their electrical charge, like whether they repel or attract water. So the AI groups those 20 amino acids into 7 distinct clusters based strictly on their physical volume and their dipoles, which is the separation of electrical charges.
Got it.
Then it looks at how those clusters appear in sequences of 3.
Oh, I see. So instead of just saying, well, we have this much carbon, it's tracking the sequence of size and electricity. Like mapping out a neighborhood by saying, here's a bulky, negatively charged house next to a small, positive one.
That is exactly it. And by calculating all the possible 3-part combinations of those 7 clusters, we add another 343 dimensions to our mathematical fingerprint.
Wow. Okay. That's a lot of combinations.
And finally, we have autocovariance. This tracks the spatial spacing. It measures how the physical properties of one amino acid affect another one located further down the chain.
So distance matters.
Very much so. And that adds the final 210 dimensions.
Okay, so you add the 20 ingredients, the 343 size and charge sequences, and the 210 spatial relationships together, and boom, you get this 573-dimensional profile.
You've got it.
But having a highly detailed profile is just the setup. How does the AI actually learn to spot the interaction cliff?
This is where Module 1 comes in. It's called constrained discrepancy learning.
Constrained discrepancy learning.
Think of the AI's training process like a bouncer at a club trying to spot fake IDs. Normally, an AI might look at the whole ID, the picture, the address, the height, and say, A, looks close enough to the real thing.
Which is exactly the AlphaFold problem we talked about.
Yeah.
It looks at the macro picture and lets the mutant into the club.
Right. To stop that, the researchers designed a specific loss function. That's the math that punishes the AI when it's wrong. They called it LCD.
Yeah, LCD.
This function calculates the embedding level distance, like the literal geometric distance in that 573-dimensional space between the normal interaction pair and the mutated pair.
Oh, so it forces the AI to look at the gap between them.
Exactly. It acts like a boss telling that bouncer, I don't care if the photo matches, I only want you to look at the holographic seal.
Right, zoom in.
And if you tell me the fake seal and the real seal are the same, you're fired. If the AI tries to take the lazy route and treat the wild type and the mutant as identical because only one amino acid changed, this loss function heavily penalizes it.
That's brilliant. But wait, even with the bouncer looking closely, how does it avoid the language model problem, where the single typo just gets lost in the thousands of normal letters?
That is Module 2, mutation site encoding.
Okay.
So eSIG-Net does utilize an underlying language model, a 650 million parameter version of ESM2.
Right.
But instead of letting the model average out the entire sequence, eSIG-Net performs a surgical extraction. It pulls a massive 1280-dimensional vector that represents only the specific site of the mutation. Oh, wow. So it's isolating the typo completely.
It isolates it,
Right.
and then feeds that specific vector through a channel-wise self-attention module.
What does that do?
This allows the AI to deeply understand the syntax and evolutionary context of just that one altered spot, completely ignoring the noise of the rest of the protein.
Okay, let me make sure I'm synthesizing this architecture correctly for you listening before we talk about how it actually performed.
Sure.
First, the AI converts the protein into a 573-dimensional profile tracking its size, charge, and spacing.
Yes.
Then, it uses a 1280-dimension magnifying glass to isolate the exact site of the mutation. And finally, it trains the system using a ruthless bouncer that mathematically punishes the AI if it fails to explicitly map the difference between the normal and mutated states.
That is exactly how it works. And once it has those two pieces, the discrepancy distance and the magnified mutation site, it concatenates them.
It glues them together. Right. And it feeds them into a discriminator. This is a multi-layer perceptron, essentially the judge that makes the final binary call. Is the interaction broken or is it stable?
Okay.
And to make sure this judge isn't just memorizing past cases, they apply a dropout rate of 0.3, meaning 30% of the neural network randomly shuts off during training.
It's like forcing the judge to make a ruling while occasionally covering up parts of the brief. It forces the system to learn the underlying logic, not just memorize the data.
Exactly.
Let's do another quick recap. eSIG-Net translates proteins into math using volume, dipoles, and spacing. It forces the AI to fixate on the exact spot of the mutation and trains it by punishing it for ignoring the differences.
Yes.
So listener, think about that constrained discrepancy learning concept. How could focusing exclusively on the difference between two states rather than the states themselves change how you solve problems in your own life?
It's a great paradigm shift, but, having an elegant architecture is great, but does it actually beat the benchmarks?
Right. Let's get to the actual proof. Because the testing phase sounded brutal.
It was. They used 5-fold cross-validation.
Explain that for us.
This is where you divide your data set into 5 chunks. You train the AI on 4 of them, and then test it on the 5th, completely unseen chunk.
Okay.
You rotate this 5 times. It makes it mathematically impossible for the AI to cheat by just recognizing data it's already seen.
And they threw 2 very different data sets at it, right?
They did. The first was a disease mutation data set, about 1,600 samples of known mutations that cause diseases across 220 genes.
Right.
The second was a population variant data set from the general public, also around 1,600 samples. But that second data set is incredibly tricky for an AI.
Why is that one harder?
Because it's highly imbalanced. In the general population data, only 16% of those variants actually cause a perturbation that breaks the protein interaction.
Oh, I see.
The vast majority do absolutely nothing. If your AI is prone to false positives, or if it just randomly guesses based on finding a mutation, it will fail spectacularly here.
So how did the bouncer and magnifying glass approach actually score?
eSIG-Net hit an accuracy of roughly 85% on the disease dataset, with an area under the curve, or AUC, of 0.91.
Wow.
For context, 1.0 is a perfect diagnostic tool. 1.91 is elite.
And what about the competition?
The absolute best sequence-based competitor they tested it against, a model called SDNN, only managed 63% accuracy and an AUC of 0.73.
That is a staggering 22% leap in accuracy over the state-of-the-art.
Massive.
And what about the structural tools? We mentioned AlphaFold-derived tools earlier. Did it beat them?
Completely. The researchers paired AlphaFold multimer with specialized mutation tools like Mutabrine2 and Beat Music. Every single one of those structure-based pipelines hovered around a dismal 60% accuracy.
eSIG-Net just humiliated them.
Truly.
But whenever I see a complex AI system with multiple moving parts, I always wonder if one specific piece of code is doing all the heavy lifting and the rest is just window dressing.
Yeah.
How do we know the discrepancy learning and the mutation magnifier are actually necessary?
Well, to prove that, the researchers conducted an ablation study.
Okay.
Think of it like playing Jenga with the AI's code. You start pulling out fundamental blocks to see when the tower collapses.
I love that analogy.
When they removed the specialized modules and only used the standard ESM language model, accuracy plummeted to 69%.
Oh, wow. So the foundation alone isn't enough to hold up the performance.
Right. Then they put the mutation site magnifying glass block back in. Accuracy bumped up to 75%.
Okay.
And
Gotcha.
finally, they added the constrained discrepancy block back in, the bouncer that forces the AI to focus on the difference. And the accuracy jumped to that final 85%.
That is amazing.
It proves mathematically that every single piece of this architecture is vital.
So, time for another recap. Tested against both disease-causing and normal population mutations, eSIG-Net outperformed state-of-the-art tools by over 20% in accuracy. It proved that sequence-based discrepancy learning beats trying to map 3D structures for single mutations. So, for you listening, which part of the ablation study stands out to you the most? Does it surprise you how much standard AI models failed on this specific task?
It definitely surprised a lot of people in the field. But the most exciting part of this paper isn't the benchmarking. No, it's what happens when you take this tool out of the computer science lab and apply it to real, agonizing biological mysteries.
Yes, let's get into the clinical applications. Because there is a phenomenon in genetics that sounds like a paradox called pleiotropism.
Pleiotropism is a massive clinical headache.
It really is.
It's when different mutations in the exact same gene cause entirely different diseases in a patient.
Which is so confusing.
Right. If you just look at the gene, it makes no sense. The gene is broken, so why is patient A getting a muscle disease and patient B is getting a neurological one?
The paper highlights the TPM3 gene case study.
Yeah.
This really crystallized the power of eSIG-Net for me.
Yeah, TPM3 codes for a protein involved in your muscle structure. The researchers mapped 2 different typos in this single gene.
Okay.
The first mutation, called L100M, causes a disease known as fiber-type disproportion myopathy.
Quite a mouthful.
Yeah, but when they ran this through eSIG-Net, the AI successfully predicted that this specific typo destroys the protein's interaction with a completely different molecule called HSF2, which is critical for regenerating muscle tissue.
Okay, so typo number 1 breaks the regeneration pathway.
Exactly. But the second mutation, M9R, causes a totally different disease called pneumonaline myopathy.
Okay, so what did the AI say about that one?
eSIG-Net looked at that typo and predicted that it keeps the interaction with HSF2 perfectly intact.
Wow.
Instead, the AI mapped how it alters a totally different downstream network. 1 gene, 2 different microscopic typos, 2 different severed relationships, resulting in 2 entirely distinct human diseases.
That is precision medicine at a level we haven't seen before. And they didn't just stop at rare muscle diseases, right?
No, they didn't.
They threw this at the CUQ8A gene, where one mutation causes severe cerebellar ataxia, while a different mutation in the same gene does absolutely nothing to the patient.
Right, totally asymptomatic.
And eSIG-Net accurately mapped the broken network in the sick patient and confirmed the healthy network in the asymptomatic one.
And we can scale this up even further. The researchers took eSIG-Net's interaction predictions and cross-referenced them with actual transcriptomic data, the RNA readouts from the Cancer Genome Atlas.
And that's a huge data set, right?
Massive. It covers over 11,000 patients.
Wow. From a single amino acid to an 11,000-person cohort.
Exactly. They discovered that the expression levels of the specific protein pairs predicted by eSIG-Net were directly correlated with patient survival rates and even predicted how well melanoma patients would respond to immunotherapy.
So to bring this all together for a final recap, we have an AI that can not only decode the mystery of why one gene causes multiple diseases, but it can distinguish between a deadly variant and a harmless one. And it can map protein networks well enough to forecast cancer survival.
It's incredible. But as with all scientific leaps, we do have to look at the guardrails.
True.
eSIG-Net is powerful, but it has distinct limitations in its current form.
Let's play devil's advocate then. Where does this tool fall short?
First, there's a trade-off for its speed. To process these massive data sets quickly, eSIG-Net uses those biostatistical embeddings we talked about rather than running full multiple sequence alignments, or MSAs.
Right, an MSA compares a sequence against thousands of evolutionary ancestors.
Exactly. And by skipping that, the AI loses some deep, long-term evolutionary context.
So it's prioritizing immediate biochemical physics over deep evolutionary history.
Right. And more importantly, it lacks cellular context.
What do you mean?
eSIG-Net predicts the biophysical favorability of an interaction. It tells you if the lock and key still fit mechanically, but it doesn't account for tissue specificity.
No, I see.
A mutation might destroy an interaction that is absolutely vital in your liver tissue, but the AI doesn't know if you're currently looking at a brain cell where that protein isn't even turned on.
So it knows the mechanics, but not the geography of the body.
Precisely. It's an interaction predictor, not a holistic cellular simulator.
Even with those limitations, though, the implications here are massive.
Mm-hmm.
We started this deep dive talking about the frustration of the VUS, the variant of unknown significance.
Yeah, the great unknown.
For decades, the only way to decode those typos was through grueling, expensive wet lab experiments like deep mutational scanning.
Right, you literally had to breed cells and watch them die in a petri dish just to see what the mutation did.
And now we have an architecture capable of performing deep in silico mutagenesis. We can test these mutations inside a computer at scale across the entire genome.
It's a completely new era.
It makes you wonder about the future of medicine. So, listener, think about this. If we can now take an AI language model, teach it to hyperfixate on microscopic discrepancies, and confidently translate the entire protein code of human genetic variation overnight,
Yeah.
what happens to the concept of an incurable genetic mystery? Are we finally crossing the threshold where biology is no longer a physical science constrained by test tubes, but a pure information decoding problem? That is definitely something to mull over.